Looks good.
Great. So I will presenting one of my chapters of my thesis. I will be speaking in Spanish. So everyone that speaks in English, please turn the captions on. Bueno, por lo tanto, voy a hablar sobre un capítulo que denominamos patrones genómicos variables en dos zonas híbridas independientes de Caballito del Diablo. Una de las consecuencias que ha tenido el cambio climático actual ha sido el de modificar el rango de distribución de las especies. Conforme las condiciones de ambiente y temperatura han cambiado en una escala global, las poblaciones han tenido que modificar su rango de distribución, típicamente siguiendo las condiciones a las que están adaptadas. En ocasiones, esto puede poner en contacto a dos especies filogenéticamente cercanas, formando zonas de simpatría. Cuando esto ocurre y los organismos tienen bajas barreras presigóticas, esto puede llevar a eventos de hibridación. Nosotros sabemos que la hibridación en poblaciones naturales puede tener una diversidad de consecuencias. Puede, por ejemplo, provocar el reforzamiento del aislamiento reproductivo, es decir, que incrementen las barreras presigóticas para finalizar el proceso de especiación. Puede provocar la extinción de una de las especies, introgresión o intercambio genético, la formación de zonas híbridas o incluso la aparición de una tercera especie mediante la especiación híbrida. Sin embargo, lo que apenas estamos conociendo son los factores que determinan que cuando interactúan dos especies, entonces se produzca una u otra de estas diversas consecuencias evolutivas. Una de las formas más prácticas para estudiar la hibridación y sus consecuencias es mediante el estudio de las zonas híbridas. Las zonas híbridas tradicionalmente las categorizamos en dos grupos. Las zonas híbridas clinales, que se definen porque hay una distribución clara de cada una de las especies parentales, con una zona intermedia donde hay un gradiente entre cada una de las especies y donde las frecuencias alélicas están altamente correlacionadas con la distancia geográfica, pero también están las zonas híbridas en mosaico, donde las especies parentales se distribuyen en parches y observamos poblaciones de una de las especies, poblaciones de la otra y poblaciones donde interactúan e hibridizan. Estas zonas híbridas en mosaico pueden ser especialmente útiles para estudiar la hibridación y sus consecuencias, ya que podemos tomar cada uno de los sitios de contacto como distintas repeticiones geográficas, evaluar qué consecuencias evolutivas está ocurriendo en cada una de ellas y posteriormente asociar a diferencias en estas consecuencias a factores intrínsecos o extrínsecos propios de cada una de nuestras localidades de muestreo. Además, otra consecuencia que tiene la hibridación durante estos cambios en los rangos de distribución es que facilita el rango de distribución de las especies. Cuando una especie presenta una expansión en su rango de distribución, típicamente en los sitios donde está colonizando, puede presentar baja diversidad genética y bajo tamaño efectivo poblacional, producto de procesos de cuello de botella o efectos fundadores. Sin embargo, si se encuentra con otra especie, entonces la hibridación puede funcionar como para nutrir a la primera especie, aumentando así su diversidad genética y su tamaño efectivo. Un modelo natural que en las últimas décadas se ha vuelto especialmente importante en el estudio de las zonas híbridas es el de los caballitos del diablo, Elegans y Graelsi, que se distribuyen en España. Elegans habitaba antes de 1900 de Francia hacia el este de Europa, pero a partir del cambio climático tuvo la capacidad de colonizar España y formar una zona de contacto con Graelsi. En España, estudios previos que hemos realizado nos han permitido categorizar dos zonas híbridas. La del noroeste, que se encuentra principalmente en Galicia y es una zona híbrida pequeña y aislada del resto de España, y la zona híbrida central, que es una zona híbrida grande en expansión y que llega hasta las Islas Baleáricas. Aquí estamos especialmente interesados en estudiar dos regiones. La región híbrida central joven, que representa el punto de avance que Elegans ha tenido en esta expansión de su rango de distribución, y la región híbrida vieja del oeste, donde ambas especies han estado conviviendo desde 1980. Por lo tanto, utilizando este sistema, queremos comparar la dinámica evolutiva de dos zonas híbridas independientes para saber qué papel juegan los factores intrínsecos y extrínsecos en moldear la composición genotípica y la introgresión de las zonas híbridas, así como qué papel juegan los eventos de hibridación como facilitadores en el aumento en el rango de distribución de las especies. Para esto visitamos ambas regiones híbridas y realizamos muestreos para estudios genéticos en los cuales en las zonas de simpatría intentamos que nuestros muestreos representaran la frecuencia relativa de cada una de las especies en cada una de las localidades. Pero también visitamos localidades alopátricas de cada una de las especies que nos pudieran servir como referencia. Con estos muestreos hicimos extracciones de ADN, construcción de librerías RADSEQ y secuenciación Illumina. Con lo cual, después ensamblamos RADTAGs y realizamos un llamado de SNPs y realizamos la identificación de SNPs diagnósticos, los cuales definimos como marcadores que estuvieran fijos para un alelo en las poblaciones alopátricas de una especie y simultáneamente para otro alelo en las poblaciones alopátricas de la otra. Entonces, recopilamos 5,702 SNPs totales y 381 diagnósticos. Con los diagnósticos realizamos la estimación del índice híbrido, que es el porcentaje de alelos heredado de cada especie, y la heterocigosidad, su porcentaje de sitios heterocigotos. Utilizando ambas variables, categorizamos a los individuos en distintas categorías de admixia y estimamos las frecuencias que estas categorías tenían en las localidades muestreadas. Con los SNPs totales hicimos análisis de estructura y de diversidad genética. Nuestros resultados de estructura genética nos confirman que efectivamente existe hibridación en estas zonas. Pueden ver en azul el clúster de elegans y en amarillo el de Graelsi, y organismos híbridos con admixia intermedia. Pero también identificamos que elegans está en un proceso de subestructuración y que elegans en la zona híbrida del oeste pertenece a un clúster distinto a las zonas alopátricas. Nosotros explicamos esto por eventos distintos durante el proceso de colonización de elegans. Seguramente elegans pudo haber llegado a Galicia acarreado por viento y después pudo haber quedado aislado del resto de España por la cordillera Cantábrica. Mientras tanto, elegans en la zona central pudo haber llegado por un modelo semejante al Stepping Stone, siguiendo cuerpos de agua nuevos que han sido creados para uso agrícola y que permiten que exista un flujo genético entre las poblaciones de la zona central con las de alopatría. Además, identificamos que los individuos, los patrones de introgresión difieren entre las zonas híbridas. Mientras en la zona central observamos una alta frecuencia de híbridos de primera generación, en la zona del oeste identificamos individuos con una variada proporción de admixia. Además, al evaluar los organismos introgresados, es decir, aquellos que tienen una alta fracción genética de una especie, pero algunos alelos de la otra, identificamos que la introgresión ha sido unidireccional en la zona central y bidireccional en la zona del oeste. Finalmente, cuando nosotros evaluamos la frecuencia de cada una de estas categorías de admixia por localidad, identificamos dos patrones interesantes. El primero es que existe una alta variabilidad en una escala local entre estas categorías de admixia, pero además identificamos que cada zona híbrida se comporta de forma distinta. La zona central presenta una distribución bimodal donde existe una alta frecuencia de cada uno de los organismos parentales y algunos organismos híbridos de primera generación, mientras que la zona del oeste sigue el patrón del enjambre híbrido, donde tenemos este grupo de organismos con variadas proporciones de admixia. La teoría predice que estas diferencias pueden deberse a distintas barreras reproductivas. Para explicar estas diferencias tan amplias que hay en una escala local entre las localidades, nosotros lo adjudicamos a un complejo proceso metapoblacional caracterizado por eventos de colonización, extinción y recolonización en la zona. Tal es el ejemplo del Louro, que tiene este patrón tan interesante. Aquí, en esta localidad, hubo en el 2010 una extinción por salificación. Después hubo recolonización y después encontramos ambas especies. Es importante saber que en esta zona la hibridación en simpatría ocurre solo en una dirección, machos de elegans con hembras de Graelsi. Y típicamente elegans es la especie rara al ser la especie invasora. Sin embargo, localmente puede aumentar en frecuencia por estos procesos de colonización. En Louro, elegans tuvo una frecuencia inicial muy alta y por la dirección en la que ocurre la hibridación se explica este patrón tan interesante de introgresión. Además, realizamos medidas preliminares de las barreras reproductivas entre estas especies utilizando cruzas experimentales, tanto de poblaciones alopátricas como de poblaciones simpátricas, evidenciando procesos sumamente interesantes. Por ejemplo, pueden observar cómo la hibridación en alopatría ocurre en una dirección, en un tipo de cruza, mientras que en simpatría ocurre en la dirección opuesta. Nosotros explicamos este proceso por un reforzamiento asimétrico del aislamiento reproductivo y que esta dirección es la que explica que puedan darse el patrón de enjambre híbrido que les mostré anteriormente. Pero además, si nosotros evaluamos la adecuación que tienen los híbridos en las siguientes generaciones, evidenciamos que en simpatría los híbridos son fértiles y que pueden formarse híbridos de segunda y tercera generación, lo cual no ocurre en alopatría, ya que solamente llegan a híbridos de segunda generación. Explicamos esto por procesos de purga de incompatibilidades posigóticas Bateson-Dobzhansky-Müller, producto de la hibridación y del flujo genético que tiene presencia en simpatría, pero que no ocurre en alopatría. Finalmente, al realizar estimaciones de diversidad genética, observamos que la diversidad genética en ambas especies, medida como diversidad nucleotídica heterocigosis, se ha mantenido alta en las zonas de simpatría comparado con las poblaciones de alopatría, lo cual refleja que efectivamente la hibridación está nutriendo genéticamente a Iznura elegans para mantener su diversidad alta y evitar estos procesos de efecto fundador. Por lo tanto, concluimos que la dinámica evolutiva de estas especies difiere de forma importante entre las dos zonas híbridas. Las zonas híbridas en mosaico son particularmente útiles para estudiar las consecuencias de la hibridación. Los patrones de introgresión de Iznura elegans e Iznura graelsi son explicados tanto por factores intrínsecos, como es la dirección del aislamiento reproductivo, como por factores extrínsecos, como es la proporción de especies, el tiempo desde la colonización y los eventos de recolonización. Y finalmente, que la hibridación ha amortiguado la pérdida de diversidad genética de Iznura elegans durante su evento de expansión, evitando procesos de cuellos de botella y de efecto fundador. Finalmente, no me queda más que agradecer a mi instituto, el Instituto de Ecología y al CONACYT, por la beca que me dio, así como al comité de tesis que me apoyó durante la elaboración de este artículo. La doctora Rosana, la doctora Yana, el doctor Enrique y el maestro Emanuel Villafán. And I want to thank you especially the Evolution 2023 organizers, which allow us to explain and to talk about our research in Spanish in an effort to make a more inclusive science. Thank you.
Thanks, Luis. Much appreciated. Does anyone have any questions? We have about 1 minute. Anyone? Oh, there's a question in the chat.
Okay.
Let's see.
Las poblaciones invasoras de elegans son... ok. Bueno, esa es buena pregunta. Justo es lo que queremos poner a prueba porque elegans tiene una distribución tanto en Francia, pero también en Inglaterra. Entonces, lo que queremos confirmar es de dónde vienen los elegans, cuál es el origen que ha tenido, y es una de las cosas que vamos a investigar posteriormente.
Okay, great. Thanks, Luis.
Yeah.
Our next speaker is Melissa Wilson, who I believe is here now. If she wants to share screen, if she can, let's see. Perfect.
Oh, great. Thank you. Can you see the screen all right?
Yep, looks good.
Great, thank you. Oh, great. Yeah, I also want to thank the organizers for the opportunity to speak and that I get to be in such a varied session. And so I am going to talk about something a bit different from everyone in this session in particular, but I appreciate you all being here. So this is our effort to design and implement a completely online and asynchronous research experience for undergraduates using genomics as our project question. So if you're not too familiar with CURES, these are course-based research experiences. And the pilot that we ran in fall 2023 was an elective for our ASU online students. So I'm at Arizona State University, and when I started, we had no online students, and then we grew to 1,000 and 2,000, and now we have over 3,500 online biology majors at ASU. And one of the things that we saw that was really lacking was the opportunity for our online students to engage in research. And with the kind of research that my lab does, which is entirely computational, it lends itself really well to this kind of experience. And so what we designed was something where in the first 7.5 weeks of a semester, they take a prerequisite course, this high enrollment computing for research, where they learn command line programming. And then a second semester, which is the intensive research experience. And we had 15 complete that, and we say 13 completed in good faith because I'll talk about a pre and a post assessment, and we didn't grade them on that. They just got graded completely 100% if they completed it. And so we analyzed 13 students here. So what was this based on? It was based on research that's done in my lab. And so we study sex differences, sex chromosome evolution broadly, and we had just submitted and had accepted a paper studying sex differences in the placenta. And this is a volcano plot showing sex differences between placentas collected from XX offspring or XY. So you might not know, and our students, most of them didn't know, that the placenta is primarily the genotype of the offspring. So you can have very large sex differences in gene expression in the placenta, and these can underlie sex differences in health and placental disease. But as is often the case in genomics, we test one, maybe two sets of parameter values, and we run with it. And so our question for this research experience was, how does our trimming parameters affect our inference of sex differences in expression in this placenta data set? And so we're going to work through that with the students. And so we divided this into 3 major areas: one, learning about the biology; one, learning about coding; and another, learning professional development. The students would learn if they were part of a lab. Oops. And this was really a team effort. So my lab studies sex differences. We collected the data for the original project. A staff scientist in the lab, Seema Pleasure, was really primarily responsible for putting together learning materials related to learning coding, our directory structure, and implementation of the analysis. And a master student in the lab who successfully defended recently, congratulations, Danny. Danny Allered was responsible for helping assemble the learning materials, making them all accessible to individuals with hearing impairment. We made sure everything was screen reader friendly also. And then we designed it to be like a lab. So we met once a week with a lab meeting, which we posted and had transcribed for anyone who couldn't attend, right? Because it's totally asynchronous. And we based our topics around the prior week's challenges that students would address to us. We also had writing hour, where students would come and set goals and regroup with us as a team. And then we had 2 additional times per week where students could come and troubleshoot like you would in lab, where you know that your lab mates are available. So they could come and troubleshoot with each other or with the instructional team. We did 2 main assessments. We did a quantitative and a qualitative assessment. The quantitative was this pre and post assessment that we asked them questions about their knowledge about the biology of the experiment we were doing, about their knowledge about coding and their knowledge about the professional development skills. And then we also asked some Likert questions about their comfort level with different research competencies. So their comfort level with coding, their comfort level with writing. Those weren't visible to the students, and they weren't graded on how many they got correct or not. And then we also did qualitative weekly progress reports. And these were designed entirely based on what I do in my lab. So every week people describe concepts and what they learned, their accomplishments that week. They discuss their challenges and, sorry, typo, and how they address them for the week. So that's really going to be a key take home here is that we ask students to self-assess their challenges and what they did to try to address it, and then define their goals for the next week, which were largely based within the cure, so they didn't have to define them too far, unless they were looking at, re-upping some skills that they felt they were a little deficient in. So here I'm going to talk briefly about how students did in the course. So overall, we see that this 7.5-week online cure increased student understanding of all the concepts broadly. So this is the pre and post assessment, pre in green and the post assessment in orange, showing broad improvement in nearly everyone. And we measured Cohen's D effect size here, and we're at 0.7964. So we're just a hair shy of a large effect size, showing that in an online asynchronous setting, we really can have large effects on student learning outcomes. We divided these then. The top is biology, with the green is the pre-test and the orange is the post-test. And these are violin plots of the student scores. And so on the left, you can see the scores are slightly lower, and then they squish up. You see the distribution squishes up towards the top. So we have higher scores for the post-test when we divide it. We see the biggest improvement here when we divide it by each question type in their biology knowledge. We do see improvement in their coding knowledge, and a little less improvement in their overall understanding of the professional development, meaning they probably came in understanding what a paper is, what authorship is. Some of these concepts we were going through, they were probably introduced to those a bit more than they were the particular biology or coding in this research experience. We also then divided each of those questions. So I showed you this is the first time we've run this. This is a pilot. So we looked at every single one of those 18 questions, those pre and post assessments, and we looked at the normalized learning gains. And this normalized learning gains essentially normalize the improvement for each student based on where they started. And so you can see that for most of them, we're having quite a large learning gain for on average across our students. And we had a couple questions that were made us scratch our heads, and I'm not going to go into them too much, but I want to talk about one in particular. So Q8 was a coding question, and it had to do with how you define R, and I realized we were getting too in the weeds, and it didn't matter. So we're chucking that one out, and we're going to come up with a different coding assessment question. But question 4 in the biology was really wild. And to me, this was interesting because so on the top is the pre-test answers and what proportion of students got it correct. And the bottom is the post-test and what proportion of students got it correct. And this was a question largely to get at student understanding of RNA and the difference between RNA and DNA and how stable RNA is. And if I can take just a second to go back to our question was, how does trimming parameters affect RNA inference expression levels differentiated between male and female placentas? And the results, which I didn't actually tell you all here, is that we found it didn't matter. It didn't matter how strict or how conservative you were being. You could trim your data to the high heavens, or you could not trim it at all. And we got the exact same genes being differentially expressed in every single scenario that we looked at. And we thought it was a bug at first, but we went through it all and it wasn't. It's absolutely true. And so that's really exciting to find out. But I think that led to a misunderstanding that RNA is stable, and in here, maybe more stable than DNA. And so although students did worse on this question, in fact, more people got it wrong afterwards. I think that has to do with actual consequence of them understanding the project and what we found. Okay, so I'm also going to walk through then students becoming more aware of their own knowledge. So this is their comfort level. I'm going to show the pre and the post test for a variety of different questions that we asked about their comfort. So either very uncomfortable, uncomfortable, neutral, comfortable, or very comfortable with this skill. And so with things like Linux, they had taken a prerequisite class where they were introduced to the command line. And then we largely used R in this class. We don't really see much of a shift in their comfort with Linux. In online collaboration, the course was based in Slack. So they're required to post code, questions, comments, respond to each other in Slack. Sorry, just checking the chat. And so we do see them becoming more comfortable there. And in our program, we see, people that we have nobody who's uncomfortable afterwards. But really where we see a big difference actually is people got a bit more uncomfortable, both with reading and writing papers. And so this suggests possibly a Dunning-Kruger effect where students were really overestimating their ability to read complex bioinformatics articles, interpret them and the methods, as well as to write these kinds of papers, because we saw a downshift in both of those. So I think that's telling and important and not necessarily a bad thing. We have those weekly progress reports where students will report their challenges and how they address them. And what we see is shifts between week 1 here on the left, moving to module 7 or week 7 on the right. And so largely coding is the major challenge for most students throughout. And then coding tapers off as we work on writing the paper, peer reviewing and working together. And then we also see blue, these personal challenges are quite low at the beginning, and then they start to really tick up. More than a quarter of the students having significant personal challenges affecting this. I should also say the class overlapped with Thanksgiving on week 6. And so it's just something to keep in mind as we're designing the cure going forward in 7.5 weeks. Holidays, family obligations can really impact our students' ability to participate and make significant advances in their research. The last was, I want to talk about our use of Slack. And so within Slack, you can get measurements out. So one of the measurements is how many days is a student active on Slack? And Danny had this wonderful idea. So she was thinking that perhaps students that were more active on Slack would be those that were the highest achieving, so the top achieving students. And so these are just binned into the top achieving moderate in blue and the lowest achieving students based on the normalized learning gains. And then the contribution is high contribution, moderate or low. And we don't really see any correlation between those that are on Slack very often and having the highest normalized learning gains. But what we do see is a significant correlation between contributing to the Slack. So just being on Slack is not enough, but students who felt comfortable enough to contribute either questions or comments on other people's posts did have the highest normalized learning gains. It's not entirely clear how one could use this information to really bring up the normalized learning gains of some other students, because there's many factors that affect whether a student contributes to Slack, whether they feel comfortable commenting, whether they feel comfortable posting their questions. And I personally don't think that if we just require them to post more, that would actually have a significant change here. So of this, 7 of the 13 students continued on in the lab. They continue on in a variety of different projects, some on this project to finish it up, some on other projects. And 2 others are interested in contributing to authors on a final publication once we get the final results of the project. We're gonna finalize our assessments here of the online cure. We're going to make a course source resource for this online cure that we're going to share with everybody if you want it. And then hopefully finalize the actual research project by the end of this summer, submit the research publication, and then design our 2nd generation online asynchronous cure that I'll run this coming fall. So that I really want to thank Danny and Seema, who without their enthusiasm, this would not have come about. NSF funded this. We had an excellent instructional designer, London, who helped with making sure that we implemented all of our materials in an online framework that was accessible to our students and all the students who participated in the cure. So I'll stop there. And
Thank you, Melissa. That was awesome. Any questions?
I see there's one here.
Yep.
When I did a collective regarding students' knowledge of the nature of science. So we didn't ask about the nature of science. I think that's a great idea. Definitely we could do that. And we have history of science and history of philosophy and nature of science as required courses. So we'd also want to assess whether they took those courses, which we could definitely do going forward. We just wanted to pilot and see if we could we do anything and be successful at all. And I think this is suggesting we really can. We just have to think really carefully about the questions and how we engage with the students.
All right, thank you. So in the interest of time, we should move on. The next person up is Matthew Glassnap.
All right, and are my slides displaying okay?
Looks good.
Okay, all right. Hi, everyone. Great talk so far. My name is Matt. I'm a PhD student in evolutionary biology at UC Santa Cruz, and I study hybridization and introgression among North Pacific sea urchins. So advances in genome sequencing have revealed that many species hybridize with close relatives and share alleles through introgression. And introgression is the transfer of alleles between species through hybridization and backcrossing. Introgression studies gained a bunch of attention after it was discovered that roughly 1 to 4% of the modern human genome was derived from introgression with Neanderthals, which are an extinct human species. There's been a recent emergence of work demonstrating that introgression often leads to adaptation by natural selection. I have some examples here, including warfarin resistance in mice, coat color in snowshoe hares, fast growth in the black cottonwood, and wing patterning among heliconius butterflies, which is a classic example of Müllerian mimicry. However, little is known about hybridization and introgression among marine invertebrates, and especially the broadcast spawners. In these groups, fertilization takes place externally in the water column. And because broadcast spawners typically lack pre-mating behavioral and mechanical reproductive barriers, such as courtship or mate choice, we might expect hybridization and introgression to be common among these groups. If sympatric species are spawning simultaneously, there should be considerable potential for hybrid matings. Recent studies indicate that introgression in broadcast spawners might be common, but only a few groups have been studied so far, including the acropora corals, the Siona ascidians or sea squirts, the Haliotis abalones, and the Middleist mussels. It's unknown whether these groups are representative of other broadcast spawning groups, such as the echinoderms. I study the North Pacific sea urchin family Strongylocentrotidae, and it's currently believed that the species in this family have not experienced introgression due to strong incompatibilities between the sperm and eggs of different species. And so to determine whether these gametic incompatibilities have been an effective barrier to introgression, we use whole genome sequencing data to test for historical introgression among these species. Specifically, we asked, has introgression occurred within the Strongylocentrotid family? If so, which genes have been exchanged between species? And how commonly has introgression been adaptive? The first method we applied to test for introgression was the Patterson's D statistic, which is also called the ABA-BABA test. So this method tests for a genome-wide imbalance in the counts of biallelic site patterns that are consistent with the two possible discordant topologies in a quartet or a rooted triplet. So I'll walk you through what that means. Shown here is a rooted triplet where O represents the outgroup and P1, P2, and P3 stand for populations or species 1 through 3. If we look at biallelic sites in the genome and assign the outgroup as having the ancestral allele A, then there are 3 possible patterns. The first pattern is concordant with the species tree, where P1 and P2 share the derived allele B. The second pattern, called ABA, is discordant with the species tree, as P2 and P3 share the derived allele. And this topology is representative of introgression between P2 and P3. The final possible site pattern, BABA, is discordant with the species tree as well, with P1 and P3 sharing the derived allele. And this topology is consistent with introgression between P1 and P3. Under the null hypothesis of no gene flow or introgression, we'd expect equal counts of ABA and BABA site patterns, and we can test for deviations away from this expectation using the D statistic, which is represented as the number of ABA sites minus the number of BABA sites divided by the sum of both site patterns. So a D value greater than 0 would indicate an excess of ABA site patterns in introgression between P2 and P3. And a D value less than 0 would indicate an excess of BABA site patterns in introgression between P1 and P3. The results of the ABA-BABA test revealed really widespread introgression across this family. I'll label the results on the tree here with arrows connecting the taxa, showing support for introgression. So the first set indicates introgression between H. pulchrimus and each of the youngest 4 Strongylocentrotus species. The second set indicates introgression between S. purpuratus and each of the youngest 3 Strongylocentrotus species. There's also support for introgression between S. intermedius and S. pallidus, between S. pallidus and S. strobosciensis, and between P. depressus and M. franciscanus. So this is a lot of introgression, but in cases where a species shows introgression with several other species that form a monophyletic group, it might be more parsimonious to assume that introgression occurred between that species and the common ancestor of the monophyletic group, which would be an internal branch in the phylogeny. So for example, it might be more parsimonious to assume that introgression occurred between H. pulchrimus and the common ancestor of S. intermedius, S. pallidus, S. fragilis, and S. strobosciensis, rather than with each of them independently. And similarly, since S. purpuratus had significant tests with each of the 3 youngest Strongylocentrotus taxa, it might be more likely that S. purpuratus and the common ancestor of these 3 experienced introgression. So this results in a total of at least 5 introgression events that likely occurred during the diversification of this family. Unfortunately, the hypothesis of introgression with ancestral lineages is directly testable using phylogenetic network software, such as PhyloNet or PhyloNetworks. A phylogenetic network is essentially a phylogenetic tree with horizontal reticulation edges that represent discrete introgression events. I'll show you a hypothetical example of a phylogenetic network with a reticulation edge indicating introgression between P1 and P3. So the blue edge here indicates that some proportion of the loci in P1 were inherited from hybridization with P3. And these reticulation edges can connect both ancestral lineages and extant taxa. So to construct a phylogenetic network, we created multiple sequence alignments for thousands of protein coding genes and reconstructed gene trees using phylogenetic software. The gene tree topologies are encoded as Newick strings shown at the bottom, and these are used as the input to PhyloNet. PhyloNet uses distributions of gene trees to estimate phylogenetic networks using maximum likelihood. And so using PhyloNet's infer network ML program, we inferred maximum likelihood phylogenetic networks with 1, 2, and 3 reticulation edges. And the output, an example of an output from PhyloNet is shown at the bottom. Again, it's encoded in Newick string format. And in the network shown here, there's a horizontal reticulation edge in red labeled H1 that connects S. fragilis and S. purpuratus. So for the results, the 1 reticulation edge phylogenetic network with the highest likelihood indicated introgression from S. purpuratus into S. fragilis. This also happened to be the D statistic with the highest magnitude. And the number shown here is the inheritance probability, which represents the proportion of sampled genes estimated to have been inherited through introgression. The network with the second highest likelihood score specified introgression from the common ancestor of S. pallidus, S. fragilis, and S. strobosciensis into S. purpuratus, which is consistent with the D statistic results that indicated introgression between S. purpuratus and each of these 3 youngest Strongylocentrotus taxa. The 2 reticulation edge network added an additional reticulation between S. intermedius and S. pallidus, which was also a significant D statistic result. And finally, the 3 reticulation edge network implied introgression from the common ancestor of the 4 youngest Strongylocentrotus taxa into H. pulchrimus, which was consistent with that D statistic result that indicated introgression between H. pulchrimus in each of the 4 youngest Strongylocentrotus taxa. So the phylogenetic network analysis seems to confirm this scenario of introgression with ancestral lineages on internal branches in the phylogeny. The last method I'll show involved comparing the phylogenetic tree of the mitochondrial genomes to the nuclear tree. The mitochondrial phylogeny supports 3 of the previously identified introgression events. First, the introgression between S. pallidus and S. strobosciensis is supported by the two clustering together in the mitochondrial tree with 100% bootstrap support. The same goes for M. franciscanus and P. depressus, who also cluster in the mitochondrial tree with 100% bootstrap support. And finally, in agreement with introgression from the common ancestor of S. fragilis, S. strobockiensis, and S. pallidus into S. purpuratus, S. purpuratus actually groups as sister to this clade in the mitochondrial tree with 100% or 72% bootstrap support rather than S. intermedius. The analyses shown thus far reveal that introgression was common during the diversification of the strongylocentrotid urchins. This indicates that the existing gametic incompatibilities were not a complete barrier to hybridization and introgression following speciation. To dig a little bit deeper and ask which loci have actually been exchanged between species, I focused on the case of introgression between S. pallidus and S. strobockiensis, and I used a phylogenetic hidden Markov model called PhyloNet HMM that identifies regions of the genome showing hybrid ancestry. So PhyloNet HMM walks along each chromosome and locates changes in the underlying genealogy, and then outputs posterior probabilities of introgression for each SNP site in a multiple sequence alignment. The introgression tracks identified by PhyloNet HMM are shown here in blue and organized by chromosome. And by introgression tract, I mean a continuous stretch of DNA with introgressed ancestry or hybrid ancestry. I'm currently characterizing these introgressed regions to see if there are any non-random patterns associated with them. And I've also identified the genes that overlap with these regions and will be working to determine whether introgression was adaptive in any of these cases. Here's the distribution of track lengths for introgression tracks greater than 10 kilobases in length. A total of 49 million bases were declared introgressed, which was about 5% of the bases in the reference genome. So most of the track lengths are short, but that's to be expected given the high amount of recombination expected in sea urchin populations. The majority of the introgressed bases fell within intergenic or intron regions, which is consistent with the expectation that much of introgressed DNA is likely slightly deleterious and experiences negative selection. I'll show you one example of an introgression track that may have been involved in adaptation. So this plot shows the probability of introgression on the y-axis by position along a chromosome on the x-axis. And there's a big spike in the probability in the middle of the plot, representing one of the introgression tracks or introgressed segments. And right in the middle of this segment is a protein coding gene called arachidonate 5 lipoxygenase. And this gene plays a key role in the immune response by initiating inflammation and was shown to be involved in adaptation in this same family of sea urchins. The gene tree for this gene groups S. pallidus and S. strobockiensis together with 100% bootstrap support, providing further evidence for introgression. And if we include the branch lengths show that S. strobockiensis and S. pallidus sequences are very similar and quite divergent from the sequences of the other congeners. So in the future, I'll be using polymorphism data to formally test for recent selection at this gene and the numerous others that I've identified using PhyloNet HMM to see if introgression has been adaptive in any of these cases. To wrap up, introgression is much more common than previously thought and has been uncovered by our ability to sequence entire genomes. Despite well-documented gametic incompatibilities between these sea urchin species, introgression was prevalent during their diversification and may be common among other broadcast-spawning marine invertebrates. And introgression may also be an important source of adaptive genetic variation, and that's what I'll be finishing up my dissertation work on. All right, I'd like to thank my mentors for their guidance and acknowledge my funding sources, and thank all of you and the evolution organizers, and thanks everyone for being here. Let me know if you have any questions.
Thank you, Matthew. That was great. I think in the interest of time, we need to move on, but if someone has a question, they can add it to the chat or they can send a direct message to Matthew in the chat as well. So our next speaker up is Enrique Soto.
Yes. Hi, do you hear me? Hi.
Yes, we hear you,
Yes.
but I don't, there we go. Looks good.
Okay, perfect. See, I freezed. I don't know why. Okay, well, thank you so much. Thank you for being here. I'm going to talk to you a little bit about part of my work that I've been doing in my PhD, and it's about population genomic structure and diversity of these species of salamander, or as we call it, shallot. Well, as introduction, I would like to talk about a little bit about the species. Bistom altamirani is an endemic mountain mole salamander that lives in permanent streams in central Mexico, and the vegetation around the streams are pinus and avid religiosa forests. As many amphibians, as many other species, and Bistom altamirani face some threats. One of them is habitat fragmentation because agricultural modification, urbanization, invasive species, pollution because of anthropogenic activities, and also in pathogen infections such as Batrachokytium dendrobatidis that generates chytidomycosis and is a big problem in amphibians globally. Because of all of these, populations decline, particularly in this species and in others. And according to the IUCN, this species is in the list as endangered. And as you know, in this context, as you know, conservative genetics are important to reduce the risk in distinction of threatened populations, and genotypic data, microsatellites or SNPs or another markers allow us to infer a lot of information related to genetic diversity structure, endogamy and depression and inbreeding depression, gene flow and migration rate, effective population size changes, demographic history, and so on, just to mention a few. And taking all this information is going to help to take better decisions in conservation management. Particularly speaking, in Bistom altamirani, there has been some studies published that studies are about the genetic diversity in Bistom altamirani comparing with other Ambistoma species, and there is like low genetic diversity comparing with another species, and also there are some studies, particularly in population of Bistom altamirani, that have been done studying the population structure. So they have found population structure, the genetic diversity differentiation, but to our knowledge, there has not been a study that focus on analyzing the genetic diversity and structure using SNP data and taking into account more populations. So that's why we wanted to do this study. So the sampling we did in this region in Mexico, particularly speaking in the Mexico state, in the Sierra de las Cruces is called the region. And this is a plot, an example of all the sites that we sampled where we found the individuals. And of each site, we sampled this number of individuals, and we sampled in a non-invasive way, where we took a tip of the caudal tissue, and with the tissue of each individual, we did with all the different analyses and process, DNA extraction, we sequenced by ddRAD-seq, and all these later with the raw sequences. I did the quality filtering and all the risk mapping. We used the Ambistoma mexicanum reference genomes to do the mapping. Later we did the snap calling and the SNP filtering. And the next analysis, I did it all of them in R. And to identify possible sites under natural selection, I used PCR-DAT. And I did later with all the neutral SNPs, the following analysis related to genetic diversity with all these functions and programs of R, highlighted in bold, and genetic structure and some insights in migration and isolation by distance. After all this number of SNPs after the filtering, I kept only this number around 7,000. And first of all, the genetic diversity, what we found was there was significant difference between populations in heterozygotes and standardized multilocus heterozygosis. And Villacarbon was the the most diverse comparing with the other populations. And when we looked up the endogamy, the inbreeding degree, also we found significant differences. And I did the correlations between inbreeding degree and heterozygosity, standardized multilocus heterozygosity, I found a significant negative correlation. I also wanted to look at the ancestry structure. And when I did this study with a K of 2, I found the individuals assigned to one population that they were from Villa de Carbon, and the other individuals from the other populations were in another cluster. With a K of 3, there was another population differentiated, org. 4, there was a TechPan or Tech differentiated, the ancestry, and the best K value was 5. And you can see there are structure of the populations with some evidence of gen flow, plus you can see in the different clusters. Also, I did to try to see the migration rate, relative rate between the pairs of populations. And in fact, the populations that are closer, geographically speaking, have more migration rate, a higher migration rate compared with the other species. And Villa de Carbon is more far from the other population, as the migration rate is lower, as you can expect. Also, to see how different were, genetically speaking, the populations between one another, I did this neighbor joining tree, and you can see there are agrupations between the different species. Seguayan and Tecpan are more similar, and you can see here Villa de Carbon is more divergent comparing with the other populations. And I saw that also with pairwise FST heat map, when you can see the genetic structure also. And Villa de Carbon has a higher value of FST comparing with the other species. And so that's very interesting. And also to try to identify the role of geography or geographic distance, I did this correlation between geographic distance and genetic distance, and as you can see, there is a positive correlation, significantly positive correlation using the Mantel test. So, in brief, what we found, no? We found that genetic diversity and inbreeding differences, between populations, negative correlations between inbreeding degree and genetic diversity. So that's a question that I asked with these results, right? If there is there are kind of signal, there is a signal of inbreeding depression, but for this more studies are needed, more to do fitness study to corroborate this. But there is a fact that there is a diminution of genetic diversity when increased the ingredient degree and in these natural populations. Bistoma tamirani are genetically structured with evidence of gene flow, geographic distance, influence population genetic differentiation, and Villa Carbon is the most diverse. This is something that I found interesting. Villa Carbon is the most diverse and structured population when compared with other species. Maybe it's more diverse, but also the most structured. So maybe there is it's going to process of population expansion, or the effective population size expansion, or maybe where there is having genetic gen flow with another populations that we didn't sample near the locality. So there's too much to do yet, but here are some insights. And yeah, that's it. That I was really fast. Thank you for listening. I don't know if you have some questions or preguntas.
Thanks, Enrique. Any questions? You can hit the little raise hand if you have one. Well, I'll ask a quick one. Can you elaborate? I didn't quite understand the very last bullet point, what your speculative reason was for that one population being different in diversity measures?
Diversity difference?
The-
Villa Carbon.
The via carbon.
Villa Carbon, yes, it was this one. I can see because of the okay. Yeah, this one, the blue one, it was most differential structure, yeah, from the others. So, well, what I saw also, as for example, this population is the most diverse, but also is the most different genetically speaking difference that the others based on the FST, for example, right? Because here you have this population is more diverse, and also based on the pairwise FST values, also is the most structured as you can see here, and that's kind of expected in this aspect because of the geographical distance. That, as we see, there is a influence.
Thanks.
Yeah, welcome.
Any other questions? We have a little bit. Oh, there's one in the chat for you.
Oh, okay, let me see. Okay, I don't know if I can answer in Spanish or English.
You're welcome to do either, whatever you want. Whatever
Okay.
you want.
Okay, the main threats in the abistomas, in the Genomics, pues de las principales, como mencioné, es justo las amenazas de la actividad antrópica, ¿no? O sea, y particularmente la agricultura, ¿no? Es la habitación que genera mucha fragmentación del hábitat. También, pues la urbanization y pues la contaminación, ¿no? O sea, justo que les mencioné, y hemos encontrado también que estos individuos están infectados con BD, no muestran, con este patógeno, no muestran señales de enfermedad aún, pero pues es un factor importante a considerar. Sí, de nada.
Thank you. Okay, anything else? Any other questions? All right. Thank you, Enrique.
Thank you.
We have one more talk. So our next speaker, we might want to wait 1 minute before we get underway, but Danielle Drebeck is next. So if you want to share your slides and then maybe wait till right at 4:30 to get started.
Can you see the slides? Okay, starting exactly on time at 4:30. Thank you everyone for coming to this talk, and thanks to all the other presenters for the super cool talks. This has been an awesome session so far. I am really excited to share this work. This is the first time I've presented my postdoc work with Suzanne McGaugh in her lab. On convergent selection, relaxed selection, and gene loss in the PLAT1 gene related to the loss of visual acuity across vertebrates. There we go. I like to just say who has done what at the beginning of talks instead of the end. In particular, I want to acknowledge Aaron Gilbertson and Jonathan Weiss, who are the two undergraduates who did work on this project, and they Aaron did the majority of the gene loss blast work, and Jonathan did the majority of the synteny mapping for this work. And the rest of this work was a huge collaborative effort between Suzanne McGaw, Richard Borowski, Andy Glusenkamp, Ira Oroyev, and Liz Alter and Diana Arcilla, either one. I think Liz is actually here. Hi, Liz. So first, I'm going to start off by talking about gene loss and why it's important. So what can gene loss tell us about adaptation? Because traditionally we thought of looking at adaptation as something that we should really focus on positive selection to understand. But recent work using genomic data has really shown us again and again that gene degradation and gene loss can be really important means of adaptive evolution. And in particular, convergent evolution is a powerful tool for understanding the genetic basis of traits. So again, with the advent of genomics and the explosion of genomics, we started to be able to uncover the physiologies of traits because of by looking at convergent independent physiologies. And the same is true for applying that to gene loss. If we can look at convergently lost genes, we can start to understand what those genes do and what are all their pleiotropies. So we were interested in vertebrate lineages with gene losses, and in looking at this data, gene loss data generated by Erin as part of her undergraduate project, we noticed that this gene called PLAT1 was missing from all squamates. And then we further confirmed this loss with reciprocal blasts of raw reads, synteny mapping, and examination of intergenic regions for all the squamate genomes that we could get our hands on. And we confirmed that, in fact, there is a real true loss of this gene in the squamate lineage. And this is interesting because vision loss has been proposed to have occurred at several points in the squamate evolution, including at the base of this lineage. So previously, it's been thought that snakes have a fossorial ancestor, but this more recent genetic work and molecular work has shown that it really is at the across squamates and at the base of squamates. So what does PLAT do? The prevailing assumption is that there are two functions, or let me start from, there are a lot of different pleiotropic functions. So there's tumor suppression, insulin metabolism, P53 mediated apoptosis, viral translocation, and interestingly, for us, lens clarification. So PLAT1 is thought to be the main gene regulating lens clarification in fish, and PLAT3, its paralog, is supposed to, or is thought to be doing this in mice and mammals. And just really briefly, the biochemistry of the way that this works, when you form a lens, you have to basically make a big blob of tissue, and you need that tissue on the inside to be clear, so you have to kill all the cells on the inside and also lyse all of the organelles and clear them out of the way so you have a nice clear lens. And the way this works is there's a widespread damage to the membranes. When that happens, the PLAT protein, it's also been called the HRASLS, so you'll see that on some of the images. It's the same thing. That protein recruits to the membranes of organelles that are damaged. It attaches through this transmembrane region here, over here. And then it basically bursts open the membrane with this catalytic domain. And the only thing that's really important to know about that is that there are these two regions. Each of those is responsible for these two functions. So the LRAT homology domain is the lecithin retinol acetyltransferase region. That's the catalytic domain, and the transmembrane region is how it recognizes what membrane organelle, or what organelle membranes to attach to. And so that's pretty well worked out. We know where those things are in this protein. We know that PLAT1 knockouts in zebrafish cause cataracts, and we know that PLAT3 knockouts in mice cause cataracts, but there are 5 PLATs in humans and paralogs elsewhere. So we don't really know what PLAT is doing in other mammals Plat1 is doing in other mammals, fish, or vertebrates. We know it has physiology related to the eye, and we now know about this loss in squamates. So our big question was, does Plat1 degradation underlie vision loss in other blind vertebrates? So is Plat1 functionally important for vision across a wide swath of vertebrates? And to answer this question, we utilized some publicly available resources. So we used the NCBI Plat1 ortholog database. We also used Ensemble's orthology database. And we also utilized Plat1 sequence pulled from 13 novel cavefish genomes that we have been sequencing as a part of another larger project. And these are dispersed across the teleas tree. So this image is of all the sequences of plat that we had aligned for this project. And I just put this in here too, because that's a pretty small or a large image and you can't really see all the foreground taxa. So this is all the things that are, I'll use the term LVAB or cave blind fish. And that just means a low visual acuity or blind species that we're designating basically as the foreground for this approach. So what we did is we looked for signals of positive selection and relaxed selection using the HiFi suite, specifically Abisrael, busted, relax, and Emmy. In the interest of time, I can't go over the details of each of these, but if you're interested in chatting about those afterwards, I'm happy to talk the details of what those different mathematical models are parsing apart. We also looked at things like gene degradation, premature stop codons, frame shifts, and we also looked for true and complete loss. So it's really, really gone. There's nothing in the intergenic region that we can discernibly blast at all. And so without further ado, let's get to the results. So for mammals, what we found is that with no designated foreground species, so just looking at the tree without any prior hypothesis, there's no evidence for selection. When we put blind species in the foreground for Plat1, we do find that there is strong evidence for relaxed selection and some evidence for positive gene-wide selection. Because we were able to do Emmy, that designates the sites as well, we can sort of further break that down. And what we found is, so this is for all mammals, and this is the kind of gross looking protein alignment. We found 4 sites that are under positive selection. This one at the N terminus is the only one that is also designated as an evidence ratio greater of 10 for busted. So that's sort of indicating that this is the only site that is driving the signal for positive selection, with the remainder of the sites are really directing the signal for relaxed selection. And those are all in the transmembrane region or in or near the transmembrane region. So before we move on from mammals, we wanted to ask, what about Plat3? Because in physiology studies, Plat3 is indicated as the main lens clarification function for mice, at least. And so we wanted to ask whether Plat3 was also under selection or relaxed selection in blind mammals. And in fact, we recovered no evidence of positive selection among the blind mammals in Plat3, which is quite surprising. We did see some long branch lengths in Myotis and the blind golden mole, but nothing was significant. So for mammals, we saw strong evidence for relaxed selection. We saw that the locality of that signal for relaxed selection is centered at that C-terminal membrane region. And then moving on to fish, what we did for fish, you see a cave. So cave indicates a cave or a blind fish, just for short. And we have a GR, that's a gene remnants test. So for some of the fish, there were premature stop codons for which there was downstream region that was just visually by eye. We were able to identify like the pseudogenizing gene portion. So we were able to run a test with and without those gene remnants included, ostensibly asking the question of whether we could recover evidence for relaxed selection after the premature stop codon was in place. So what we found was, in fact, that again, selection is detected only when cave and blind species are in the foreground. So there's a lot of signal for selection, but overall, relaxed selection was the stronger signal, and both signals for positive and relaxed selection were increased when we included gene remnants. So something definitely also interesting going on with fish. That's not the complete story here with fish, because as if we look at the test of selection, they're blind to indels. And if we just take a really quick peek at this alignment, these are important functional regions highlighted in yellow. We can see that there are very large structural changes and all of these changes are actually in blind or cave fish. There are no large structural changes in any of the other fish. So we decided to formalize that sort of analysis, and we found that there are 6 species with really large indels, so 3 with premature stop codons and 3 with large deletions in functionally important regions. So it's significantly, cave and blind fish are significantly enriched for structural changes in Plat1. So for summary fish, what we found is the signals of relaxed and positive selection for cave and blind fish are both increased when we include gene remnants, and we did find the most convincing evidence for relaxed selection. We found that cave fish lineages are also significantly enriched for structural degradations. So coming back to the sort of bigger picture question, does Plat1 degradation underlie vision loss in other blind vertebrates? The answer we think is yes. So we did find relaxed selection in low visual acuity mammals in Plat1, but not in Plat3 as expected. We found 6 independent instances of deletions among cave fish and relaxed and positive selection on blind and cave fish lineages. We also found gene loss in all squamates, and sort of as a summary, Plat1 appears to be important as a convergent target for evolutionary vision degradation. Some important insights, if I have 1 extra minute, is that Plat1 is associated with the mammalian eye function, so it's not just Plat3. So that suggests that the lens clarification function might be possibly ancestral to Plat1, so should be followed up with some gene family function studies. And also some insights into evolutionary pleiotropy. Plat1 is lost in some cave fish, but not others. And it might be an important question to ask why. Is it the age of the lineage, or the function is different in different species, or there are gene duplications that, that cause things to have analogous function? And sort of bigger picture, here is combining these insights from gene loss and convergence and selection can provide these large scale insights into gene family function and evolution. And I think I have 1 minute for questions.
Yes, you do. Thanks, Danielle. Anyone have a question?
For a relaxed test, did you test along the leaf branch or include? So we did include all the internal ancestral branches leading to every lineage. They were, I think it's 16 species, and I want to say it was 12 independent lineages across those, because we had, 4 species that were very closely related and another 2 species that were very closely related, and like maybe 1 more off the top of my head. But yeah, we did include internal branches.